Welcome back, life science learners. Let's continue with our revision lesson on genetics. We were looking at pedigree charts before we went into the break. I know that these can be very confusing and often require a further detailed explanation. Let's take a few more questions on this and then look at another pattern of inheritance where we refer to as sex-linked inheritance. I wasn't complete with that question before we went into the break. I'd like to go back to that and unpack some of that when we look at how pedigree diagrams are interpreted. So again, I'm going to go back to this and I'm going to refer to this chart, and the question that we're going to get onto is it possible for individuals C and D to have a child with free earlobes? So guys, when we look at individuals C and D, if we remember, I illustrated this earlier on that again, the free earlobes is the dominant trait, so we refer to them as being either that or that, so homozygous or heterozygous. And the individuals that are free will have to have the recessive alleles. So essentially, these individuals have to have two recessive alleles. So knowing that these individuals are homozygous recessive, let's try and unpack the next part of the question. Is it possible for individuals C and D to have a child with free earlobes? So essentially, if we have these individuals that are crossed, no matter what happens in a genetic diagram or a pedigree or Punnett square, guys, we know that all of these individuals that are produced will always be homozygous recessive, as I've indicated in this Punnett square. So the answer would be, is it possible? The answer is no. And I know that it's not simple enough to be able to settle for no. Let's try and understand why you say no. And essentially, my explanation to this would be, guys, if you've got parents that are homozygous for a particular characteristic, and both these individuals we refer to as pure breeding, when we cross those individuals, the only possibility that we have in the next generation is the same characteristic appearing. And so in this case, when these parents are crossed, there is no other allele but the recessive alleles that can be inherited. And so essentially we would say that since C and D have attached earlobes, they have only the recessive genes, and as I mentioned, these are the recessive genes, and can therefore have no dominant gene or genes for free earlobes to be passed on to their offsprings. So it's literally impossible for that to happen. Okay, and guys, as I mentioned earlier on, the key to working out many pedigree diagrams is to identify what I refer to as the individuals that are affected or homozygous recessive, because you've got to know that when an individual has these recessive alleles, it means that both the parents have to have the recessive allele. So when we look at this individual here, which is individual F, and if we allocate that individual the recessive alleles, it's important that we recognize this fact, as that during independent assortment, each of these alleles come from the parent. And we know that this means that individual F would have inherited a recessive allele from the mother and a recessive allele from the father. And that is crucial to individual F inheriting two recessive alleles. And then that should help us to determine what the father is. And because he has free earlobes, which is the dominant trait, he has to have the heterozygous combination. So again, guys, the golden rule is to look for individuals that are affected with the condition. Again, individuals that are affected are homozygous, as I mentioned here. And use the rationale of asking yourself, where did they get those two recessive alleles from? And that means they would have got it from their parents, making their parents at least heterozygous. They could be homozygous, recessive, they could be heterozygous, but they cannot be homozygous dominant. So you will not get an individual that has that producing an offspring that has that. So that's the key. And again, you can use that same rationale to infer, like we would in this example, that this individual, which is the female, can only give all the offsprings the recessive allele, meaning that all of these offsprings have to inherit a small f from the mother. And that clearly is because she has two recessive alleles, and all that she can pass down to the next generation is the recessive allele. And that is the key to working with pedigree charts, is to first establish the individuals that are homozygous recessive, and then use them again to determine what they pass on to the next generation, or to determine what they have inherited from each of their parents. And that's the clue that I would like to leave you with as we move on to the next bit, which is looking at sex-linked inheritance. So guys, when we talk of sex-linked inheritance, again, it's important that we recognize that gender is determined by specific chromosomes. And we've looked at this in the past, where we know that individuals have two chromosomes which indicate the gender. The X and X chromosomes indicate that the individual is female, and the presence of an X and Y chromosome indicates that the individual is male. It's important that we recognize that on these chromosomes that they are genes, and these genes are inherited in a specific pattern, and we refer to that as the sex-linked inheritance. Cool. So we spoke about the X and Y being the sex chromosomes. The Y chromosome is much shorter than the X chromosome, which essentially means that it contains fewer genes. All males have a Y chromosome, and that Y chromosome is significantly smaller. On the other hand, the X chromosome is a longer and contains significantly more genes than the Y chromosome. Here's an electron micrograph indicating a comparative size between what we call the X chromosome and the Y chromosome. And essentially, the concept of a Y chromosome is derived from the appearance of this, which appears much shorter based on the position of the centromere. But we'll chat about that at some other point. So this is essentially showing you how different the X chromosome is and the number of genes that it contains, whereas the Y chromosome is significantly shorter, having fewer genes on them. When assigning alleles for sex-linked traits, the convention is to write the allele as a superscript for the sex on the sex chromosome. We're going to unpack that by referring to some characteristics that are sex-linked. It's important that you take note of a few of these genetic disorders that are sex-linked. It does help when you know these, because when you do read this in the context of an exam or test, you know immediately that this is a pattern that needs to be sex-linked. So hemophilia is one example. Color blindness. Cystic fibrosis is another example. So these are three common examples of characteristics where they are found on the X chromosome. And they follow a specific pattern, and we've got to recognize that in our next slide. So the example we're referring to is hemophilia. And we tend to use, again, the gender as well as the allele as a superscript. So essentially, you've got to indicate the X, the dominant and recessive alleles using that notation. And again, we have the Y chromosome in males that does not have the allele. So remember that this is an X-linked characteristic. Often we get learners indicating on the Y chromosome the presence of the allele, and that's wrong. Again, it's an X-linked characteristic. It can only be found in the X chromosome and not on the Y chromosome. So that would be an incorrect way of representing sex-linked characteristics. So let's get straight into trying to work through a problem. Hemophilia is a sex-linked disease caused by the presence of a recessive allele indicated using that. A normal father, and I often like to use the word unaffected. Again, the context of normal meaning hasn't been affected, unaffected. And a heterozygous mother have children. So guys, the concept here is that the parents, firstly, the male is unaffected, and that is a cross with a heterozygous female. So the female is heterozygous. And I think when you read through a question like this, it's important that we recognize early on in the question the genotypes and the phenotypes of the parents. When we refer to as a male being unaffected, we need to say that it's the male that is unaffected. Essentially, because it's sex-linked, we need to indicate the gender of the individual. And that's key to solving genetic problems using sex-linked inheritance. Again, because it is a male, we had to indicate his gender. So that's the first step. Crossed with a female, again, indicate the gender. Now we determine what the alleles involved in this. Again, the male is unaffected. There are two possibilities of males, either being affected or unaffected. So again, knowing that hemophilia is a recessive condition, it means that this male who is not affected has the dominant allele, because we noted that that's the notation for the recessive allele. It's important to recognize that the female is heterozygous, meaning that she has the dominant and the recessive allele. Again, so I've got to allocate a heterozygous combination to that. And that's fundamental to getting the rest of the problem correct. So now we've established the genotypes of the parents. So let's read the question. Represent a genetic cross to determine the possible genotypes and phenotypes of the children of the parents mentioned above. And that's a 6-mark question. Guys, again, this is a genetic cross using a genetic diagram. Let's refer back to our template and let's try and unpack that. So again, I'm going to very quickly illustrate that. Again, we had the phenotype of the parent, so it was an unaffected male. And it's important that I indicate the gender as well. Crossed with a heterozygous female. And this is the important bit now, guys, the genotypes. So the male is XY, crossed with a female, that's XX. At this point, we need to indicate the allele. So it's an unaffected male, so capital H, heterozygous female, small capital H, small h. Note that I've clearly indicated the difference between my capital letters and the smaller letters. It's important that we now form gametes and we separate them out. I often encourage my learners to circle them to indicate that these are standalone gametes that are haploid. So essentially what we're doing is we're separating out the alleles into these gametes. The next bit requires that we use the Punnett square to illustrate the possibilities that might occur in a cross of this nature. We now have the male and female gametes illustrated on either row and column. The next bit's quite easy, but you need to remember that it's important that we first cross the gender, and I'll illustrate that. So let's cross the gender, and let's do that. When we did the first cross between these two, we note that there is a dominant allele there from that parent here and a dominant allele from that parent there. Likewise, here we get the Y from this parent and we get the H dominant from that. And so I'm going to quickly wrap this up for us as we conclude this problem. And I'm going to get through to that individual who is now going to be recessive. Looks a bit messy, but we can quickly illustrate that as being one individual, female, who has that. So I've ticked that off. Notice the next female is this individual here who is heterozygous, which I've kind of now ticked off. And this helps me to be able to ensure that every individual in that Punnett square has been presented in my genotype and phenotype ratios. So what I've quickly illustrated here is the possibilities of having individuals. And finally, as we wrap this segment up, we've got to write down the phenotype ratio of the individuals. So we've got to indicate the gender, guys. So that's going to be a female that is unaffected because she has both dominant alleles. So that's an unaffected female. The next is a female, but if you look at her genotype, she's got the dominant allele and the recessive allele, making her what we call a carrier. So she's not affected by the condition, but has the recessive allele and may pass it on to the next generation. And finally, we've got the males, which we have to indicate as males, and this individual has the dominant allele being unaffected. And the last male, again, being a male that has the recessive allele, so that's going to be an affected individual. And guys, there are many ways that these questions can be asked. One could be determine the possibilities of having an offspring that is affected. And we've just established that there's one male of the 4 that could be affected, a 25% chance. Guys, you've been a fantastic audience today. Trust that you've enjoyed the interaction, that you can go and work with some of these problems confidently. Wish you well. Have a baie lekker day. See you soon.